get started. All right, what is this? And Lincoln Taste does not get the answer. So every living thing is made up of cells. And this is a beautiful example. We are all modular construction. We're all made of the same basic kind of module that's highly similar from plants to fungi to humans. And what that is a cross section of a pine needle. And it's a really beautiful example because there are cells that specialize in forming an impervious layer on the outside. There are all these cells in here where chlorophyll is that do photosynthesis. Then there's all the plumbing inside the pine needle. There are the cells that specialize in bringing water to the needle and carrying sugar away. So a really good example. And I should say that these cells in this pine needle, in their molecular complexity, is more complex than the cells in your body. Plants can do things that we can't do, many things. They can do almost everything else that we can do. Okay, so in our own bodies, of course, these are the cells that at this moment are bringing the material that you just ate for lunch inside your body. These are cells that line your intestine. There are a whole series of different kinds of cells. Perfect example of modular construction. So we should get started here, really put this in the context of the size and complexity of cells, because, you know, we all listen to these astronomers and they give us these huge numbers. Well, biological world deals in astronomical numbers. So every person is made of 100 trillion cells. 14 zeros. And every cell is an enormous world in itself. Every cell contains typically 100 trillion atoms. How can that be? Well, just think, that cell's got a copy of DNA. That DNA encodes for at least 25,000 different kinds of proteins. Many of these individual proteins are present in maybe 100,000 copies in a single cell. So this is a complex world, but it's a very tiny world. And what I'm hoping to do today is to show you sort of our best look at it in really living cells. So one way we can look at this is with an electron microscope. So, and this shows you that there are two basic kinds of cells. There's simple bacterial cells that kind of have a wall and there are no internal compartments. Even these, though, are quite complex. A typical bacterial cell may have 5,000 genes in it. And so the biochemistry in here is very complex. But most of the organisms that we can see with our eye, plants, fungi, humans, have these types of cells here that we call eukaryotic cells because they have a nucleus. And the other difference in these kinds of cells is that they have all of these internal membranes, which form a whole series of compartments, all with specialized functions. So you can kind of just, if you look at the sort of smallest dots, you can see these membranes here just barely make out little tiny black dots on them. Those black dots are the ribosomes. This is something Harry Noller worked on here at UCSC. These are the protein manufacturing machines, and every one of those black dots is a huge complex of about 80 different kinds of proteins and 4 huge RNA molecules. So getting a way to really look at this complexity has been a difficult challenge. Now, the electron microscope has wonderful features to it in that we still can't see any individual proteins. I just said that the tiniest black dot is something that has at least 80 different proteins within that black dot. But his disadvantage of the electron microscope is you have to kill the cell, you have to slice it, you have to embed it in some kind of resin, and you usually have to, all of these things are transparent, they're not colored. So to make them show up, you have to coat them with some sort of metal. So it has big pluses to it as a technique. What we'd really like to do is see it like the way we do in the movies. And we come a little bit to the stage where we can do that. I'm going to show you kind of where we stand with that today. So here is your textbook example of a cell. So, and this would apply to plants, fungi, animals. So you have the nucleus where the DNA is. Then there's a whole elaborate membrane complex whose main function is to make membranes and to make the proteins that are part of the membrane. All right. And then there are other ribosomes out here, little protein manufacturing machines that make proteins that aren't in the membrane. You have a series of rods and cables. You have the mitochondria, which basically convert your food to a molecule that can be used as the fuel for the cell, ATP. So lots of different kinds of structures in here. Well, what does it really look like? And how can we see it? All right. So the organism that I've been working with all these years here at Santa Cruz is a fungus called Neurosper crassus, quite a famous fungus in the biological world. The Nobel Prize for discovering what is the function of a gene, and that function is gene specify the structure of proteins. That was done with this organism, with Neurosper, because the genetics were so simple, you could manipulate it so simply. And so it's a nice, tough, easily manipulated, genetically modifiable organism that's been a subject of cell biology. The pictures I'm going to show you today are from these Neurosper cells. They grow very fast. This is obviously speeded up, but maybe only 30 minutes for it to grow from down here all the way up here. And you see as they grow, they lay down these cell walls. See, there's one formed there, then one formed here. It's sort of that essentially constitutes the cell in this, but each of these has a little pore, and all the material can be motivated to go up here to the growing tip. So what you see is this kind of fine granular stuff in the cell, and we know there's 25,000 different kinds of proteins in there. So what does it really look like? Okay, so there are two big innovations that have occurred in the last 20 years or so that allow us to look at cells in a way we've never been able to before. The first is the ability to basically put a colored tag on any protein in the cell that we want. And so the way this is done is pretty simple. One of the beauties of modern molecular biology is you really can do it for a few $100s in your garage. It's kind of scary, actually, what you can do. But if you want to look at a particular protein, find out where it is in the cell, for example, well, you can isolate the gene from that protein. We won't go into how that's done, but it's very straightforward technology nowadays. You've got the gene in the test tube, and you can get from another lab, genes that code these little proteins that, if you shine the right wavelength of light on them, will emit colored light. They'll emit green light, red light, orange light, blue light. They've made all different kinds now. And so in the lab, in the test tube, you just mix your protein that you want to make colored. You make the protein that makes the colored light. You fuse them together. You add an enzyme that clips them together. You mix your cells in there, and there are ways of getting this DNA to go into the cell. One way is just give it an electric shock, interestingly. And so at a low efficiency, but the way that works, your fused gene will now go into the cell. It'll go into the nucleus. The nucleus will see this as a fragment of DNA and think, there's been breakage here, and it'll incorporate into the rest of the DNA that's in that cell. And it will be treated like all the other genes and turned on and off at the appropriate time. Except that when this gene is transcribed, that means copied to be made into protein, it'll get shipped out into the nucleus, and these ribosome protein manufacturing machines will convert that into protein. And what you'll end up with is your protein of interest attached to one of these little light-producing proteins. Well, many of these, by the way, are isolated from jellyfish. Interestingly, if you've been to the aquarium, it's jellyfish glow. Some people got the proteins out of there. Great example of how basic biology, why should your tax dollars be used to study jellyfish? Because it can lead to great innovations in molecular techniques. So the ability to tag proteins so we can see that is important breakthrough number one. Now, breakthrough number two is improvements in microscopy. So we have this microscopy center here at UCSC. Part of it's up in Six Labs, the others at the Biomedical Sciences Building. And at its core, the new microscopes have several important differences. The core of the microscope really is not much different than it was 80 years ago. You know, it's a series of lenses, you have the eyepiece, and that's how you magnify the image. But the difference is, you don't just have a light bulb shining light up through your sample. You have a laser. Kind of works just like your TV screen. It sends a line back and forth across the sample. And B, instead of just eyeballing it on top, you have a very specialized, very sensitive camera. And then maybe most importantly, here is the whole software apparatus that looks at the light and it uses mathematical algorithms. You know, that little piece of light is actually probably a distortion. We'll get rid of it. The real light is just the light that's over here. And so it can process your image. The other major difference is that a good microscope years ago might have cost you $50,000, and now this one costs you $500,000. All right, and I'm going to show you an expensive one later on. All right, so with this, you can start to really see inside cells. So here's just an example. So working in the lab with this gene, a lot of the things we've done in the lab have looked at proteins that transport molecules across biological membranes. And there are a series of proteins that all look like they were calcium transporters, but we didn't know where they were in the cell. So one of them tagged it with green, and when you look in the cell, it's in these little rings. And so the question is, what are those little rings? Well, they were about the right size and shape to be the nuclei in the cell. In this particular kind of cell, there are more than one nuclei. There are multiple nuclei. So how could you find out whether those really are the nuclei? Well, you could get a protein that you know is inside the nucleus. And you could tag it with red. And so we have this red tag nuclear protein. And we put these two colored proteins in the same cell, take the green picture, take the red picture, at the same time superimpose them. And sure enough, the red protein is inside these round structures, and they are in fact the nucleus. Now, one important kind of quantitative thing to stress here is if we see this red blob, you know, I've told you, you can put this red emitting protein on another protein. Can you actually see a protein? And the answer is no. The difference between which you can see an electron microscope, or you're using electrons to illuminate it, versus here, where you're using light. Mainly, you're limited. It's very difficult to see anything that's smaller than the wavelength of light. And the wavelength of light is quite a bit bigger than protein. So this particular, let's say, blob of red here is actually the light being emitted from probably 100 or 1,000 proteins. So the resolution is a lot better, but we're still not seeing proteins. All right, and I'll give you another picture that will help you put that into some context. Okay. So the other beautiful thing about this is you don't have to kill the cell, you don't have to fix the cell. You can look in live cells. And so this is the nucleus of these cells that we're looking at, up at the tip. And you can see that this is actually growing. This is going to loop around. So here it starts. The very tip is right here. It's manufacturing new material, new cell wall, new membranes out of this tip. And then the material is moving slowly into the newly made space. And you can see some of these nuclei are dividing, where you see the long one here, this one's starting to divide. And instead of being a nice round organelle, it's becoming very elongated. Here's one that's dividing in here. And so now you really have a technology. Is there an issue with the? What's that? Okay. What we have, this is Zoom. So can I do it without getting out of screen share? Okay. Oh, in there. Yeah, I see. I see what you mean. All right. Perfect. Thank you. Okay. So it's just very cool to be able to see this live, and in color, and see the different parts of the cell. So I'm going to take you kind of on a quick tour. We saw that textbook photo of how we draw the different organelles in the cell. Well, what do they really look like? And what do the nuclei look like? Well, they look like this. All right. So we said that the nuclei are often kind of connected to part of. Okay, now. So, okay. Oh, there we go. Okay, the endoplasmic reticulum. Now, this is a set of membranes, which it turns out includes the membranes that surround the nucleus. But from the nucleus, they also have these side channels that form a structure kind of like the radiator in a car, stacks and stacks of interconnected pancakes. And their function is to make membranes and to be the site where proteins that are in membranes are located. And so when you see what this looks like, first of all, it's impressive just how much of this stuff is. The cell is just loaded with this. What you're seeing here are the envelope that surrounds the nucleus, and these other kind of less well-defined green spots are membrane fragments floating in this cell. Now, it has a wall right here, and there is a tiny little hole right at that point. And so you can see these nuclei come up here, and they squish right through that tiny hole and pop out on the other side. There's a big vacuole here. It's too big to squeeze through that hole, so it's kind of stuck in this corner of the cell. All right. So in the way cells make themselves, the gene starts out in the nucleus, and some of these proteins that get made from the gene go to the endoplasmic reticulum, incorporated into membranes. Then they go to another organelle that's called the Golgi apparatus, and it is less well defined. In fact, this is an interesting finding to us as to what it would really look like. Couldn't really identify what it was in the electron microscope, and that's because they're just kind of poorly defined little membranous patches within the cell. And then ultimately, all of this is driven by the energy produced by this organelle, the mitochondria. The mitochondria look like little worms. And if you read a little bit of biology, you know the interesting thing about the mitochondria in the cell is we knew for years that at the molecular level were very much like bacteria. And we now know that in fact, they were bacteria. They're the degenerate ancestors of bacteria that invaded the first eukaryotic cells back, many, many millions of years ago. But we have so many of them because we have this constant need for energy in our cell. And each of these little worms has thousands of these little energy generating molecular machines inside the structure. All right, so really kind of beautiful to see what they really look like. All right. And then there is some structure to the cell. After all, is it just a lumpy, flexible bag? We know that it has some rigidity, some shape to it. And part of that comes from the fact that there is effectively an internal skeleton to all cells. And it's made up of a protein that can form a rigid system of cables, fairly rigid cables, and or rods really. And these are called the microtubules. And so what's shown here is you can see all these fine lines in here that form the microtubules inside the cell. And they kind of give it a molecular skeleton around which this membrane wraps. They have a second function, though, besides this structural function. If you have molecules that are down in this end of the cell, and let's say they're little food packages that are needed up at the growing tip of the cell, how do you get something from here up to here? And the answer is there's sort of an internal railroad system. And these are the tracks, the train tracks. Another fascinating aspect of biology is that there are proteins that essentially function like little trains. They literally have one end that grabs onto one of these membrane vesicles. It's filled with some needed cargo at the tip. And at the other end, they have proteins that essentially function as legs. They're little molecular motors. I'm going to tell you about one in a bit, one called Dynein. So if you had a microtubule on the floor here, it would attach to a tubule at one end of it, and then its feet literally march along the microtubule during its cargo. Okay, and I'm gonna show you a picture that illustrates a little bit how that happens. All right, so it's just this is really cool experiments to do. I really love this in my retirement. Really gotten into this microscopy. It's such gratifying kind of experimental science because you put all this together and you put it in the microscope and you get instant results. You see it live on the screen. So what I'm going to do here in sort of a second half of my talk is talk to you about the specific project that I've been doing recently. So one of the reasons I volunteered to step in here is just a couple of weeks ago, what is probably my last paper, it just appeared, and I thought, oh, this is a chance to talk about it. So it's a little nitty-gritty detail in the great world of cell biology, but it kind of illustrates how you can use these molecular and microscopic methods to answer some sort of biological question. So what I've been working on in my career here is really this organelle within the cell called the vacuole. It's the same as the organelle that's in animal cells called the lysosome. And it is effectively the recycling center of the cell. So one of its major functions is, proteins either get damaged in the cell, or there are times when we no longer need them. And so the cell has a way of taking those proteins and putting them inside this organelle. And it's sort of like the stomach of the cell almost. It's a very acidic space because there's a pump that pumps acid into here. And these proteins are degraded into amino acids, and the amino acids can be transported out and be made into new proteins. So cells are really very efficient recycling all the materials. And this can be done for lipids. It can be done to remove phosphate molecules that are attached to proteins, etc. So there are in these organelles a number of different kinds of enzymes that are soluble in the soup inside this organelle. And then there are other kinds of proteins that are transporters or have other functions that are embedded in the membrane of this organelle. And much of our focus has been on this transporter that pumps acid into the interior, determines the acidity inside, and allows for the maximum operations of these enzymes that chew up other kinds of proteins. So our bragging point here is that this enzyme, which is present in plants, humans, fungi, has an important role in all cells, was discovered here in Santa Cruz by me, by my wife, Rusty, sitting right there, by Lincoln Days and Lincoln, sitting right there. So we had laboratories next to each other for 35 years, and figuring out the origin and structure of that enzyme was part of our career. So when we got to the point where it could finally having worked for this for 30 years, could not even put a tag on it and see where it is. Actually watch this thing. We're very eager to do it. So this vacuolar structure occurs in two different forms in this organism. So you see these great big vacuoles. I showed that endoplasmic reticulum movie where there was a big vacuole stuck in one part of the cell. And the older parts of the cell have a lot of these big round vacuoles. But up at the tip, where it's really actively growing, this same compartment takes a completely different shape, this long, thin tubules. So they look like this. This is actually a vacuolar compartment, all these tubules. And how do we know it's vacuolar? Because we find that the proteins that are in this sort of spaghetti network here are the same proteins that are back in that big round vacuole. All right, so when we first put a tag on the VATPase and looked at the tip of the cell, this is what we saw primarily, this whole tubular vacuolar system. Now, this was known to exist before. This wasn't a new discovery. It really also said that our tag was a good one because the enzyme was showing up in the place where we thought it should show up. But interestingly, made some movies of this, and here you see this kind of spaghetti of tubular vacuoles back in here. Out at the growing tip, brightly shining, were these mysterious organelles. They're kind of big and round, but normally you don't have the big and round ones up at the tip. That kind of didn't make sense. So it was really a mystery organelle. Now, people have been studying cells for 100 years. You don't find a new organelle every day, But I talked about it at meetings. Does anybody know what this is? No, nobody had really seen it before. So I had to give it a straightforward name. So I just thought I'd call it what it was. It's an unidentified fungal organelle. It's a UFO. So I spent several years trying to figure out what this UFO was. It's unidentified fungal organelle. Notice, for example, it takes funny shapes. Sometimes it looks like it has compartments. Sometimes it looks like it's open at one end. I'm sort of moving around the microscope here. Sometimes it looks like it's a nice, simple, round organelle, and other times it's very distorted. So it's quite peculiar. All right, so basically, here's one of these long fungal filaments. And if you look back in here, you see there are some fairly large, round vacuoles. And then as you move up towards the tip, you get this dense collection of tubules. And then when you get to the very tip, you see this new unidentified fungal organelle. So if I blow up the picture here, magnify it, so here are the tubular vacuoles, and then here are these interesting odd organelles. So what are they? Are they some kind of specialized vacuole? Well, one thing you could say is, all right, if they're a vacuole, I know a whole bunch of proteins that I predict will be in there. So are they in there? And got a surprising result. And that is that if I looked at proteins that were in the membrane, the ATPase, the calcium transporter, the amino acid transporter, they were all in there, consistent with it being a vacuole. But if I look for proteins that were soluble inside here, the enzymes that chew up proteins or chew up lipids, they were all missing. So it was like had half the components of a vacuole. And that was a suggestion that maybe it's a vacuole in the making. All right. So there was another peculiar aspect of it. And that is that I mentioned that sometimes in the pictures, it almost looked like it wasn't all enclosed, that it was open. I mean, does it have an inside to it? And so I could test that by using certain strains of cells produce this red pigment. It actually makes the fungi look red if they make it. And if you look on the microscope, the red pigment is inside these big vacuoles. It's inside the tubular vacuoles, but it's not inside this new UFO, organelle. Now that was very puzzling. I mean, it's a soluble molecule was behaving as though it was embedded in the membrane. Or in fact, is this just not really a round compartment? So this was the question to be solved. And so how do I explain this highly variable shape and this peculiar observation that it didn't seem to have an inside? So sometimes these UFOs would look like this, very bizarre. Sometimes they would have these sort of blisters on them. Other sides, they would be very lopsided in their structure. All right, so set out to solve that problem. So the way I approached it was, I'm not going to solve this with a $500,000 microscope. I need something good. And fortunately, we have this stem cell center, which is supported by the taxpayers of California. Thank you very much. All right, that bought a new super resolution microscope. So the physicists, clever guys that they are, have found out how to beat the problem that you can't look at anything that's shorter than the wavelength of light. And so they're called super resolution microscopes because they go beyond the resolution, what was thought to be a theoretical resolution. Basically what they do, and I cannot give you a real good explanation of the physics, but if you know the shape of the light that's going through the image, and it comes out on the other side in a shape scattered in ways that are not consistent with what it was looking at, you say, oh, some of that light was scattered and isn't really showing you the picture. We will subtract that light and we'll just show you where the light going through the real image should have been. All right, that's sort of how it works. So with this kind of microscope, and what you can do is you can, when you take a picture, it's just a narrow part of it that is in focus. If you do regular photography, you notice there's a focal plane. And so you can take a whole stack of pictures and take them in very rapid time, unless it's just photos, slice. So in this case, I've taken 40 pictures, kind of optical slices through one of these organelles from the top to the bottom. And I selected from those 40 12 to show you here. And what you see as you work your way up through this organelle, you find sections clearly that are just completely open to the outside. Nothing collects inside these in terms of soluble contents because it doesn't have an inside. All right, and then as you work up to the top, you see then it looks like it has this normal organelle, although even there, what you see is that it's very lumpy. It's not as though there's just two lines that form this kind of donut. And here's where the software comes in. Now I have all these 40 slices, and I can ask the software to put this together in a three-dimensional structure and rotate it for me. And so what does it, well, oh, I'm getting a little bit ahead of myself. So let's just look at this slice right here. Okay, and magnify that. And what it looks like is that it's kind of not like a rope, it's more like a necklace made out of beads. That the whole structure, so there's clearly spaces in between these. So the little tubules are little beads that make up this structure. And if we just took one of the beads and I draw here the appropriate sizes of the protein that I have the tag on and the appropriate thickness of the membrane that would make this little vesicle, you can see the membrane is barely visible there. It's that sort of blue-green line. That's how thick the biological membrane really is. And the little yellow dot, that's the size of how big the protein that tag really is. And so what we're seeing here in a 200 nanometer slice is the glow from all of these proteins on this membrane. So it gets you some sense of the degree of resolution. Pretty good, we're looking at something really tiny, but still, not, we can't see the proteins per se. All right, so ask the computer to put that together into a 3D image, and here's what it looks like. So in fact, it's shaped like a pouch made up of individual little round vesicles, and one end of the pouch, in fact, is open. And so just wherever you slice it, it's going to explain how sometimes it could look like it's a horseshoe shape. Now, here's another similar picture. Here I backed off a little bit so we can see multiple examples of these organelles. So if you look at this one from this angle, you can see it's open at this end. This one is clearly open at this end. And so the structure turned out to be really very different from the first thought when you looked at it, thinking, oh, it's like a balloon. It's not like a balloon. It's really made up kind of a very different and very complex structure. All right, so last aspect of this, great example of kind of serendipity. And I told you about those molecular motors that carry vesicles from one end of the cell to the other. One of these is called Dynein. And a lab that worked on Dynein had put the red tags on the parts of the Dynein that bind to these little cargo vesicles. And we went to the same meeting, work on the biology of these cells. And they said, our clumps of Dynein look like in the same size and they're in the same place as your mysterious organelle. And so here were their red tags on the Dynein. Here was our mysterious organelle. You put them in the same cell, and sure enough, these Dynein motor fragments are inside in that space in the pouch. And so here's a movie. Here are these two organelles. There's a pair of them here. And this Dynein is carrying material to the tip. So the tip is loaded with Dynein. And every once in a while, you'll see one go zipping by. That's one that's being carried along on a microtubule. So it's on the fast track going on microtubules. They're called comets, because you see the little red comets go zipping by. But other bits of the Dynein are bouncing around inside the UFO, my mysterious organelle. All right, and so we can magnify that, and we can rotate it, and we can see that in fact this pouch contains fragments of this Dynein molecule. And exactly what it's doing in there is unknown, but it probably almost certainly has to do with the fact that it has the ability to bind to little vesicles. So to wrap it up, here's the conclusion that I draw from this microscopy, that if you're making an organelle, you make this vacuole, you start out by doing it in the endoplasmic reticulum to make membranes and membrane-embedded proteins. They get a little modified, and then address is kind of put them on, take them to the vacuole, and they get put into little round vesicles. And these vesicles are aggregated or collected somehow, probably by the help of this Dynein, what has the ability to attach to these little vesicles and position them somewhere. It's like a vesicle sorting center. And then at a later stage, then the soluble enzymes that are needed inside the vacuole, they come in later and they're added to the inside of these vesicles. And then they fuse to make these tubular vacuoles. And ultimately, you get a complete organelle. So thank you very much. This is the slide I always use when I give talks in other places. I think a few of us have used this slide. It's great to say, this is the lab. Wouldn't you like to work here, you young postdoc, et cetera? We are sitting about right here. All right. Various people who have helped me. I have to acknowledge my wife, Rusty Bowman, who's been involved in all of this, and the TASES. We've worked together for years. So thank you.
Sure, Barry would be happy to answer questions.
Yes.
Can we have a little more light?
Yeah. turn on the light. Well, we'll put these up because the lights come on very slowly. Okay. Al,
Harry, you mentioned that these are common to all multi-cell organisms. What would you be, your guess be at what point those things evolve? Because they are not incorporated bacteria, right? But at what point in terms of evolution did these maybe perhaps evolve?
so the first of these eukaryotic cells with nuclei, go back. Well, let's see. I mean, like the first cells were roughly 500 million years after the advent of Earth. I think Lincoln might know this number, but probably, the Earth is 4.5 billion years old. Probably these first eukaryotic cells were about 3 billion years going back. Actually, Dave Deemer is here. Dave would know the answer to that. Is that a good guess?
You're right on the 500 million.
Okay, all right, okay. So it's really an ancient cell. There were ancient unicellular organisms way back. And then, we got the specialization and fusion that kind of led us to the complexity we see today. But, this is a fungus. One thing people don't like to hear is one thing that's come out of the gene analysis is what are the fungi most closely related to? And it's the animals, not the plants. In fact, it's kind of an aside here, been stories in the paper the last several weeks about how people are fearing a new tide of fungal infections. And that's a real problem because fungi at the cellular level are so similar to us that almost anything that will kill fungi will poison us. And so to this day, there are no real good antifungal agents because at the cellular level, we're so similar to them. Yeah. Dave.
On your vesicles,
Yeah.
and you call them vesicles, and you said the membrane about 5 nanometers thick. Well, that's the lipid bilayer,
Yeah.
right? And would it not be an explanation for the apparent hole in it, that it is the sort of circular aggregation of your labeled vacuolar ATPases? Because it looks like there's a belt of that fluorescent red material around the vesicle, just looking at it. So instead of an opening, it could be explained as well by just a place where there is no labeled material, because the lipid bilayer was labeled.
But the opening, I mean, I have a vesicle, a 250 nanometer vesicle, and that's complete membrane sphere, presumably, and those vesicles form a necklace, and the opening is in the necklace. It's not in the individual vesicle.
Oh, I see.
Yeah, so it's the next level up in its size for the opening.
One follow-up. Do
Yeah.
you know which protein in the vacuolar ATPase is actually being tagged by the fluorescent protein?
Well, we've tagged about 3 or 4 different ones. They all look the same in the microscope. Yeah.
So if I understood you right, the endoplasmic reticulum is making the membrane material.
Right.
It goes to this Golgi apparatus and gets sorted. Is that the way you said it?
When it goes to the Golgi apparatus, there are some modifications made. For example, proteins sometimes have sugars added to them. Some of that occurs, and some of those sugars might act as sort of signals for the apparatus downstream to know where to put that protein in the cell.
So the kind of quote, nervous system of the cell, where does stuff go? Is that a kind of big open problem, or is that?
Well, this
too complicated to-
it's a big area of research. It's called the intracellular trafficking problem. And how does the cell apparatus know what goes to the mitochondria and what goes to the vacuole and what goes to the plasma membrane? And we know part of the signals. For some cases, for example, there's some specific sugars with phosphates on them that are a tag, take me to the lysosome.
I see.
But it's not simple. Okay. David.
I think clearly why you had that particular shape, the pouch shape, was it such that it was the size and the volume such that you had something attaching them? It was given space that was.
Yeah.
Did you say why?
No, yeah, David asked why that shape, and I don't have a good answer for that.
You implied it with the red thing sort of.
Yeah.
Together, but
The other thing that's mysterious is that about 15% of those pouches don't have any visible Dynein. They're empty. There doesn't seem to be. However, having said that, the keyword might be visible, they could be in there in some early stage, but I don't know why it has that pouch shape. That's something, and what gives it that pouch shape? I mean, there's some structural protein that makes it form that shape, and I'd love to know it might keep me going to another paper.
you implied that the dining is like a hook.
Yeah.
Imagine the hook actually has some.
Yeah.
Thread attached to them, then you can have this tension, there's
Yeah.
things that bind, maybe you have to find that mysterious central, I'll call it the
Yeah,
core of that thing.
but Sean, the last picture where I'm rotating it with the Dynein kind of suspended in there, it doesn't seem to be in contact with anything. That's kind of mysterious too.
Right.
Although in the movie, you could see them, they're banging all around inside there. But somehow they're held in there. So, and of course, the only things we can see are the things I put tags on. It's not as though there's empty spaces in there. There's other proteins very likely in there. Nancy.
Thanks. So I've been reading that fungi help trees communicate with each other.
Ah.
Is there anything in this that is going to help us understand that communication?
Well, first of all, that topic is something that Lincoln and Lee and I and all are much involved in, and boy, I even don't want to get started on it. It involves a lot of anthropomorphic thinking, let's put it that way. But Nancy brings up a really interesting thing, that one of the great roles of fungi in the world is the soil is filled with them. They definitely bind to and have symbiotic relationships with the trees and many other organisms. It's a really difficult thing to study. You can see that if you want to study something that is in dirt, how do you look at it? How do you dig it up? So my answer, Nancy, is it's really poorly understood. It's important, but we just don't know much about those organisms because they're so tough to study. Okay. Okay, I think we've come to the end. Well, thank you very much, folks.
One more thing, and we'll put our hands in Jim Clifford's.
I've been volunteered to take a picture of them. And the group photo, like in camp. Maybe a sports team. And right outside here, there's steps.